that particular protein, the first relay protein in this pathway, in most cells is a protein called RAS, R-A-S. And I'm not exactly sure where that name came from. I seem to recall that R might be for rat. I think it was first discovered in rats, but they find it in lots of different organisms, including human beings. Okay, so there's the growth factor. It binds to the growth factor receptor, and that activates the RAS protein. And so now the RAS protein is a kinase, and it's going to add phosphates to the next relay protein to activate it. That, and then usually that next relay protein is a kinase, and it's going to kinase the next relay protein. Here's kind of a close-up of that. Here's a review question. What kind of kinase is this? Tyrosine kinase. Yeah, the little T there is a tyrosine amino acid. And so any kinases that specialize in adding phosphates to tyrosine amino acids are tyrosine kinases. And so now that relay protein is activated, and it's going to go activate the next relay protein, which activates the next relay protein, on down the pathway until we finally activate the cellular response enzymes, which in this case are the S-phase enzymes. Now, let me turn on the lights here for a second. There's a little blob of text right here, which I don't really have a digital cartoon for, but I want to draw it on the board. Many of these relay proteins involved in mitosis are kinases, meaning they activate the next relay protein by phosphorylating it, just like I showed you there. But some of them operate by a different mechanism that I want to explain. They're called transcription factors. So here is the cell that we're talking about. And let's say this is relay protein X. It's some relay protein in this pathway that we're talking about. And so when relay protein X is activated, its job is to activate relay protein Y, which is the next relay protein in this pathway. Where is relay protein Y? Well, it hasn't been made yet. Remember, proteins get made when in the nucleus their gene is transcribed into an mRNA, and then their mRNA leaves the nucleus and gets translated by ribosomes into the protein. Well, in some cases, protein Y isn't even there yet. And so what can happen is when relay protein X is activated, it goes into the nucleus, and it causes the nucleus to transcribe relay protein Y. So it's relay protein Y's gene. And relay protein X will actually bind to that gene and promote the cell to transcribe it to a messenger RNA. And so now a messenger RNA for relay protein Y gets made, and then a ribosome gets hold of that and translates it into relay protein Y. And so now relay protein Y is a good active protein and can carry on the signal transduction pathway. So notice relay protein X didn't kinase relay protein Y to make it an active protein. Relay protein X went into the nucleus and caused relay protein Y to be made. But now that relay protein Y is made, it can carry on with the pathway. And so that type of thing is called a transcription factor. You would say that relay protein X is a transcription factor because it helps in the transcription of a gene, in this case relay protein Y. And so, not to confuse the issue here, most relay proteins in this mitosis pathway are kinases, but some of them, like this one I'm diagramming here, are transcription factors. But nevertheless, it's a pathway. Nevertheless, each relay protein in some way leads to the activation of the next relay protein. All right. Yeah, go ahead.
So relay protein X in that situation, is that reused or is it?
I believe it is reused, that after it goes into the nucleus, it promotes the gene transcription of the next relay protein gene. Then I think it would leave the nucleus and go back into the cytoplasm, but it would shut itself off. A lot of these relay proteins will automatically turn themselves off, so you don't get stuck in mitosis. That's a good observation. Anyway, so this relay protein activates the next relay protein, usually by kinasing it, and then that one activates the S-phase enzymes, which, when they're activated, are what move the cell past the checkpoint, and so now it's in S-phase, and around the cell cycle we go. But eventually, like you just pointed out, they automatically turn themselves off, and so the cell goes back into GO unless the growth factor is still present. If the growth factor was still present, then you'd keep getting reactivation of the pathway over and over again, and then the cell would keep growing. All right, and I think I pointed this out when we talked about signal transduction pathways. Here I'm only showing 3 relay proteins in the pathway, but that's an oversimplification. It's not uncommon for there to be dozens and dozens of relay proteins in the pathway. Okay, and one more point about all of this. Well, actually, I'll tell you that other point in just a second. There's a name for all these proteins that are involved in these mitosis pathways, and they're called the proto-oncogenes. Well, actually, the term technically refers to the genes for these proteins. So the genes for the receptor, and the genes for this RAS protein, and the genes for all the relay proteins, and in fact, the genes for the S-phase enzymes themselves are called proto-oncogenes. The gene for any of these proteins can be called a proto-oncogene. Including the receptor, RAS, and all the relay proteins in the S-phase enzymes themselves. And I guess I've talked about this already, that the mitosis is very tightly controlled because the body doesn't want the cell to get stuck in mitosis and keep growing and growing and growing, because that's in fact what cancer is. And so the cell only makes the growth factor when it wants the cell to be dividing, and as soon as the cell has done enough divisions, it stops making the growth factor, and that means that all these active proteins in the cell will automatically inactivate themselves. And there's actually another layer of protection. Even if the growth factor is present, if the dividing cells start to contact surrounding cells, in other words, if they've divided enough so that they've already filled in the space they're supposed to fill in, they'll automatically stop dividing. That's called contact inhibition. And as I said, it even overrides the presence of the growth factors. Here's a demonstration where they took some fibroblast cells and grew them in culture, which means in a laboratory dish. And they gave them all the nutrients they need, and they've given them the growth factor that they need, so of course they start dividing. But notice that they only divide up to a point. Once they start touching each other, then that contact inhibition comes in, and so even then they ignore the growth factor and just stop dividing. Now, if you were to scoop out some of these, like this, then at least for these ones in this area, there's no longer contact inhibition because they have an edge that's not touching. So then they will start to go into mitosis again. But again, it's a very controlled thing, and they'll only divide enough to fill in the missing area, and then the contact inhibition kicks in again, and they'll stop themselves.
So the cancer, is it that there's an unlimited or unregulated amount of growth factor, or is it?
Not normally. What happens, well, it's kind of the next section I was going to talk about, so let's just, I'll answer your question in just a minute. All right, so yeah, that's a good lead-in. Let's talk about when this process goes wrong and the cells get stuck in mitosis. That is what cancer is. Cancer cells are your own body cells that have become stuck in mitosis, so they divide in a continuous and uncontrolled manner. So they look just like your own cells, they are your own cells, but they're your own cells that are stuck in mitosis. There's some illustrations here I'll show you regarding cancer, and these are from using breast cancer as an example, but it really, the same concepts apply to any kind of cancer in the body. Shows some cancer cells that are grown in cell culture. So you can see they don't have contact inhibition. They'll keep dividing and dividing and just dogpile on top of each other because they've lost contact inhibition. And they've also lost the need for growth factors. Even with no growth factors, they'll just keep growing and growing and growing. But here's what I was saying about using breast cancer as an example. If some of the cells in the breast become cancerous, then they start dividing. They don't need growth factors. They don't have any contact inhibition. They just grow and grow and grow. And the clump of rapidly dividing cancer cells is called a tumor. And so it gets larger and larger. And the tumors tend to disrupt the function of whatever organ they're in. You know, organs are very complex things with many different tissues working in harmony with each other. And if suddenly you get this big tumor growing, it's going to upset the structure of that organ and decrease its ability to function correctly. And what's even worse is some of the cancer cells can metastasize, which means spread throughout the body. Some of the cells in the original tumor can break off, and then they could travel in blood vessels and other tubes in the body, and they become essentially seeds. They travel to other organs and start new tumors there. And then as these new tumors grow, cells from those tumors can break off and travel to other organs in the body and spread the cancer from one organ to another.
So when this happens with cancer patients, how is it that chemotherapy works?
The chemotherapy stops cells from dividing. It's essentially poisons for cells. And the idea is that the cancer cells are the most actively dividing ones in the body. And so by poisoning mitosis, you'll be stopping the cancer cells. But unfortunately, there are other cells in the body that are supposed to be dividing, like the cells that cause your hair to grow have to constantly divide. And so people on chemotherapy will lose their hair often. And the cells in your bone marrow that make new blood cells are also constantly dividing. So people undergoing chemotherapy sometimes get anemia because they don't have enough blood cells anymore. Yes, yeah. Anyway, so the tumors can spread to different organs. This is a person's liver who died of some form of cancer. And liver is supposed to be a very smooth pink organ, but you can see these whitish globs all over it. These are tumors that have spread from some other body part and disrupted the structure and function of the liver. And likewise, if you look at other organs in this person's body, like their brain and their kidneys, those would also be filled with tumors. And so what kills a person from cancer is the tumors get into so many organs and disrupt their function. The body just loses the ability to function correctly, and eventually the person can die of it. Louis?
Some time ago on the news, I seen that I guess there's a T-cell leukemia.
I'm not sure. I don't know if I saw that article. So they said that they had cured leukemia, which is a type of cancer.
Yeah.
Okay.
I seen that on the news.
University
in Western States.
I don't know. I'd have to see it and see the details of it. It could be that they found a specific way to target the chemotherapy to just T cells. Or I don't know. I'd have to see it. If you get a chance, point the article out to me. Okay. Go ahead.
So when metastasis happens, the cancer cells spread. So one cancer cell spread and lodge itself somewhere among some other tissue. Did that cancer cell itself starts to grow, right?
Yes.
And it's not that it infects the other cells, it just...
No, that's right.
Single cell.
Right. It doesn't infect the other cells. It itself starts to grow. So you get new tumors in some different organ than the cancer already started in.
Has it developed a way to fully find all tumors in the body? Before they spread?
To find them, I don't think they have a perfect way of doing it. What they do a lot of times is look at things called lymph nodes, which are little structures in your body where some of the tubes merge. And so if cancer is spreading from one area of the body to another, a lot of the cancer cells get stuck in those lymph nodes for a while. So they look at the lymph nodes, and if they see the cancer is in them, that in the lymph nodes, it indicates that it has spread, that the cancer is spreading. So that's not a good sign.
Are you like able to estimate where the cancer is originating from? Since, like,
They do have some way, because if they find tumors throughout the person's body, they can normally say, oh, this started as breast cancer or this started as lung cancer. So I think if they look at the cells under the microscope, they can somehow figure out whether they started as lung cancer cells or bone cancer cells or whatever. All right, so at a molecular level, the cancer is caused by mutations in these genes that we've been talking about that are involved in mitosis. And to be a little bit more specific, think of these proto-oncogenes. Remember, the proto-oncogenes were the genes for any of these proteins in this pathway, like the